Good morning and afternoon for some. My name is Amy Blythe, and I'm with Trihydro Corporation, and I'd like to welcome you to today's webinar, Practical Science and Reality of PFAS Measurements, Pragmatic Evaluation of Your PFAS Data Against EPA MCLs. We're excited to get started today, but first we have a couple housekeeping items. First of all sessions are being recorded and will be available after the webinar. We'll plan to send those out to you within a week of from today, and for all registrants, those people will receive that email. All attendees are in listen-only mode. If you have questions, please submit them via your Q&A panel on the Teams platform, and we'll get to them at our during our time together today. If we don't end up getting to them, we'd be happy to follow up with our answers via email after the webinar. With that, let's get started. Andrew?
Thank you, Amy, and welcome to all of you. My name is Andrew Powlitz, and I'm a regulatory toxicologist with the Trihydro Corporation. And then extending the introduction given by Amy, wanted to provide context to today's webinar. That is to help those entities subject to the PFAS MCL rule understand the key elements of the compliance requirements as they pertain to analytical data resulting from drinking water testing. Particularly, the topics selected include a closer look at the analytical methodology and instrumentation, as well as data quality checks on the results provided by laboratories. And then the overall objective is to inform the user, i.e. the regulatory community, what to look for and what measures to take, and perhaps how to communicate with the laboratory to ensure receipt of defensible and quality results for the purpose of compliance with the MCL rule. Next slide, please. Let's take a look at the basic MCL rule elements. There are two sets of requirements. One is the MCL, or the enforceable maximum contaminant level, that shall consider implementation feasibility, and the other being the MCL goals, which are non-enforceable and do not consider rule implementation costs or feasibility of treatment options. However, both have risk-based components intended to protect the public from PFAS exposure through drinking water. The rule names 6 PFAS substances of various chain length, ranging from 4-carbon, such as PFAS substance, to as long as the 8-chain substances, such as PFOS and PFOA. Next slide. Let's take a look at the overall Safe Drinking Water Act MCL development pathway in context of PFAS. The process started back in 2009 with the contaminant list designation. Then we had the UCMR3 in 2012, and then regulatory determination in 2021, with the final rule promulgation earlier this year. One thing to keep in mind looking at this process is that every 6 years, there's a rule review and revision process, which for the PFAS rule will be taking place in 2030, when additional PFAS substances might be added or revisions made to ensure continued protection of the public. Next slide. This table not only lists on the right-hand side column the current MCLs expressed in parts per trillion MCLGs, but also in a hazard index term. And this table also outlines the evolution of federal drinking water limits since 2009, with the trend starting high or non-existent, then lower still in 2022 with parts per quadrillion levels, and coming back to parts per trillion currently, plus the hazard index. Next slide. Moving along in the interest of time, just wanted to show you the equation for the hazard index concept, which provides summation of the relative contribution to the overall hazard index for each of the 4 non-carcinogenic PFAS substances. And this is really unusual because typically we have compound by compound or individual compound MCL numbers. But in this case, we have a hazard index approach. And this assumes additivity of non-cancer effects. Next slide, moving along. In terms of implementation, public water systems are to monitor for the 6 listed compounds by 2027, specifically using methods 537.1 and 533. And those will be covered by the speakers on this webinar in detail. And then public notification is required if MCLs are exceeded, and the treatment will be needed to the MCL levels or alternate water supply provided by 2029. There are some concerns with the laboratory capacity by the sheer number of samples that will be taken and the number of public water systems affected by this rule. In addition, also is with the promulgated MCL, now this is a point of departure, so to speak, for the state rules, which shall not be less protective than the federal limits. Next slide. Quick preview of what the MCL compliance may look like. Table 2 lists the results of the water testing across the US. And based on the current data available, a significant number of public water systems will likely encounter individual MCL exceedances. Next slide. And this table shows the potential rate of exceedances based on, again, current results of the hazard index departures. Keep in mind that the UCMR5 data collection is ongoing, and complete results are expected by 2026. And this is in advance of the general deadline for monitoring commencement by PWSs. And slide 9 is my last line, summarizing the main points, and then underscoring and highlighting the importance of ensuring defensible quality PFAS results, which the next speakers will provide more details. So thank you very much. The next slide provides my contact information should you need additional guidance or information on the MCL rule implementation. So this concludes my presentation, and I'd like to welcome the next speaker.
Oh, come on. You know, the minute I get something good going here,
We can see you now, Jen.
now,
Oh, your camera went back off. So if you want to put your webcam on. There you are.
okay, can you hear me?
Yes, we can.
Okay, well, good, thank you. I apologize for that. I had to end up getting a new computer and a new operating system and a bunch of other stuff. So, really, we're going to be doing HPLC on a lot of different waters, different cleanliness, different things like that. But the biggest problem seems to be, having done some of this prior to the EPA introducing their regulations, it's cleanliness and contamination. This is where the lab most often falls down. Next slide, please. In starting to look at this and look at the clean water and what the people can do, there's a lot of companies that state they can clean water down to undetectable levels. Every time I hear that, I always ask one question, and definitely discuss it with them prior to working with them. What is the detection limit? If it's 100 ppm, it isn't going to do much good. It could be undetected because it's lower than that. If it's 1 part per trillion, hey, bonus. So always just ask that. And I've seen that happen so many times in environmental science and in other areas of science as well. When you're doing a run and you've set up your analysis, always do periodic method blanks. So that's water you've taken through the entire process, but you didn't have any sample with it. That's going to really address the cleanliness of your instrument, your sample prep methodology, how that works. Clean water blanks is going to just take a look at, is the water you're using contaminated with your target chemistry? And then non-inject blanks, that's looking at, you're not doing your injector, you're not doing anything like that. That's looking more at your tubes that are going to your mass spec or your detection methodology. And so that's going to help you troubleshoot if anything goes wrong. I ran a group. It was called Special Projects. We had at times up to about 30 chemists, most of which were doctorates, doing special things for the company that I worked for, things that didn't fall into any other area. And as part of that, we had to do some very odd testing things of things like, we had TFA, that we had to be at or below a part per trillion, So this isn't quite our first go around. So in July of 2000, I gave a talk at the University of North Dakota at my technical director's asking on the quantitation methodology of the lower molecular weight perfluorinated acids at 1 part per trillion. And we ended up, we could go further than that. This was kind of similar to the background levels in the water that was used was, we had to clean the water to below that 1 part per trillion level. And to do that, and this also now works for, we later on validated it for the bigger perfluorinated carboxylates, and then the perfluorinated sulfonates, and a number of other odd duct chemistries that are related to that or that may degrade to form those chemistries. We cleaned the water as follows. We had 2 of the big carbon tanks, so they're about the size of a gas tank, the gas tanks you hook up to your GCMS. They were virgin carbon. We put them there. We put them first. So it went into one, filtered out, came into the second one. Then the second, after it went through the second, we did this in conjunction with millipore systems. And so millipore has a little bit of records on this. They're still looking for other stuff. Excuse me. Okay, so yeah, we completed with the 2 carbon tanks, and then we, to improve the peak shape, we ended up using an ion exchange technology. So we took all the positive cations out of there and replaced it with sodium. The reason we did that is it just really cleaned up and gave us much better peak shape. We had better signal to noise, we had better peak shape, and that helped drive that quantitation limit down. So now we have our GAC, we have an anion exchange, and then the final treatment system, we just use 2 of the smaller millipores. Maybe they're 2 feet high, something 18, 20 inches, within the UV light to shine on it. And we ran it through one of those, and then we ran it through another one of those. And then we monitored our water. So when it was coming out of the GAC system and it was starting to get dirty, we'd remove the first tank, the first tank that the water encountered, take the second tank, make that the first tank now, and put a new clean tank there. similar things with the filters on the millipore systems. What we did is we could detect, but not really quantify reliably at quite a bit lower than the 1 part per trillion. So 1 part per trillion, was definitely a nice target. And this can help you do your analysis at these low part per trillion levels just by cleaning your water and getting good peak shape. Okay, next slide, please. In addition to clean water, we needed a cleaner lab, and this is kind of where we set up. And the reason it started is one person had the high level standard of the potassium salt of PFOS. They would come trotting through the lab, and it went poof, and we contaminated the entire lab. And because of our air handling system and the way it was working, samples got contaminated. It was a mess. We ended up having to clean a lot of stuff that we never thought we would clean. And after that, we put in an area that would be for high level standard sample prep. So you have your bottle, you have your sample in it, and in this case, a white powder. You pull it out, you do your measurements in there. Everything's in there. That thing is sealed up, and they get the first primary standard into liquid, so that there isn't any air-to-air contamination. And then we started taking that a little further. We found that HEPA filter was better because sometimes we ended up with air coming out of the lab that had low levels contaminating this area, and then we should HEPA filter from this area into other areas. We ended up with an area for the dilution of the high level standards because accidents happen. Things fall on the floors, people step on them, things along that line. So we had a separate area. We had a separate area for preparation and the curve preparation area. Preparing curves, it ended up, we actually showed that one guy in the lab, and this goes more to the next point, one guy in the lab would be running these samples, and have maybe 120 samples in the sample tray, and he'd get most of the way through, and he'd start pulling the vials out and recapping them in the lab. And he'd do this for like maybe an hour and a half. And by the time he got it done, we were showing contamination on neighboring instruments and on his own. So we put it in there so the sample, everything was prepped in a separate area. And we always made sure that people weren't opening besides instruments. It saved time, yes, but it did contaminated. And then we also recommend that the areas are cleaned floor to ceiling daily. The high level standard room, yes, definitely. The high level dilution, yes, definitely. Some of the other ones, you might let slide. It just depends. But have those blanks in there I talked about on the prior slide so that you can check and really make sure that everything's clean. Next slide, please. Your containers and your vials, your sample containers, and I think this is going to be gone through in greater detail, and associated lids should be non-metallic. Use only clean and certified sample containers. Don't use dirty ones, and make sure that when they say clean, it's down to that level of less than a part per trillion of PFAS chemistries. Always check 2 or 3 empty containers, 2 or 3 unused lids in each lot by adding just pure water and analyzing for PFAS, your targets you're after. After samples, containers sealed, labeled with the date on them, placed in secure refrigerated storage. That was always me. We did a lot of GLP work. And inevitably, you have a sample train of 150 samples, and somewhere in there, that auto sampler would pick up one and throw it across the room, or drop it on the floor, or spill it. Something would happen. And if we had the sample, or the non-inject, so I always made up an extra duplicate set of samples, and I always saved the ones I had analyzed because auto samplers can go nuts sometimes. Once the samples are analyzed, you replace them in a vial container and hold them for reanalysis if necessary. So that was some other lab practices we have. Now we're going to talk about why we're seeing PFAS in a lot of strange places. So next slide, please. Next slide, please. Did I go?
changed and it did,
Hello?
yes, it did advance.
Oh,
The contamination slide is up right now.
okay, so I'm sorry. Um, Contamination. Uh, PFAS chemistries are widely used. Where I was working, they called it, and they also called it at pixie dust level. They put pixie dust to quantities of PFAS everywhere, and it helped a lot of things work. So they help release molded plastic parts from molds. So someone has this hot plastic, it has a cold mode here, they put a little PFAS in it, then they pour it in there, and then it helps it pull out, and you got a nice clean little mold. PFAS chemistries help polymers and metal sheets move on rollers that transport products from point to point in the factory. Examples: plastic bags, aluminum foils, textiles, natural synthetic rubbers, rubbers inside of vial caps. Sometimes they're contaminated with PFAS. That's because they were running them through here and then they used a stamp and they stamped them out and put them in there. PFAS chemistries are often sprayed on non-finished good to inhibit static. So as this stuff is running across these rollers or and we'll have some more of that later, or the thing that's rolling across it is sprayed with dilute PFAS. That rolled manufacturing is the highest volume manufacturing in the world, virtually in every country. They're rolling everything around. It's cheaper, it's easier, and as long as there's PFAS around, things aren't sticking. They spray it to inhibit oxidation on some products. So dilute PFAS in water will inhibit the degradation, so it shows up. They're used on thousands, and I think the actual number is unclear, but I think it's more like tens of thousands of different products. Anywhere that they have a roller, they've got a spray bottle of dilute PFAS. They had a spray bottle of dilute PFAS, and they were going, and we still run into those. Most of the use data you can find, there's a nice article out there, it's referenced below. They pull a lot of uses out of there. So if you're curious why it's spread everywhere in the world, you can kind of look at the number of products that are put in there. Next slide, please. Do I have a next slide? Oh, there we go. This is an example of a roll coater. And what happens is that steel roll there, the largest one, the first one, they'll put a little bit of glue underneath, and then that plastic runs by and they put glue on it. However, the bottom side is coated with PFAS to keep it from sticking. So that's a coating operation. There's other operations, just moving this stuff around, bagging it, so treat every film you see, whether it's an aluminum foil or a plastic saran wrap or a bag, those plastic sacks, treat them as contaminated because they probably are. Next slide, I believe I have one more. So they also use this, they call it web handling. So they spray it on things that this is just moving everything and start processing. They spray with very dilute PFAS to help the film from sticking during manufacture. Process includes winding, so they're winding things up. What they call rolling, so they're just kind of moving it around. When they're inspecting it, like this little guy here is that great big roll of paper. Who would have thought they've been spraying paper with PFAS, and they use that paper in boxes to make stuff not move around. So when you get an Amazon box, it's got a bunch of paper in there. Good chance that paper has PFAS on it from your Amazon box. Cutting, they do a lot of cutting. So if they want to make a big piece of paper, a smaller roll of paper, they cut it, and then there's your PFAS showing up. So the bottom line on contamination is you got to treat everything as if it's contaminated. You check anything that touches that sample prior to analysis. That includes your hands, that includes you, that includes everything that gets it touched. When you're doing that, all of a sudden this analysis becomes quite a bit easier than what most people think it is. So thank you. That's kind of the end of what I was talking about, I think.
Well, hello, everyone. You've heard a couple of great talks so far. Andrew sort of introducing the regulatory aspect of it, and Amy, or Jennifer rather, has been quite good about introducing all the ways that you can contaminate samples, whether it's in the field or in the lab. I'll mention a few more of those along with the talk. But today, let me introduce myself for a brief minute. My name is Ken Rosnack. I neglected to put my email address on here, but it's pretty easy. It's Ken_Rosnack@waters.com. So pretty simple. Our email address is firstname_last@waters.com. I am a market development manager with Waters, been here for 25 years, it's hard to imagine, and been involved in mass spec in some fashion for, I call it 4 decades because that sounds a smaller number than 40 years. So been around the block a bit, using instruments probably that most don't even know about these days. But I work with a number of colleagues within Waters, and the information and the data that you see today has come from them. They don't allow me to touch instruments anymore because I have a habit of breaking it. And I know a few colleagues are listening in on this, or may in the future. So at this point in time, let's continue on with the talk. And it's not letting me control the slides. And maybe now, let's see. All right, so firstly, let me talk about Waters Corporation. Many of you may have heard us. We've been in business for at least the past 65 plus years, starting off in a building that was the jailhouse in Milford, Massachusetts. So our headquarters is located there. We are a $3 billion company with nearly 8,000 people worldwide in over 35 plus countries. We look at a variety of systems, and I'll go into mainly some of those shortly, with service, informatics, consumables, and then all the hardware and software that goes along with it. We work in a variety of different market spaces. Environmental is where we're going to talk today, but we also deal with food, pharmaceutical, clinical market spaces as well. So let's see if I can control it at this point. Yes, I can. Thanks, Amy. And a big thanks to Amy, because she's the one that has set up this entire program and dealt with those of us in it. So a big shout out to you. So Waters is has a broad and expanding technology portfolio. Again, starting off way back when, most known as a liquid chromatography company, and that has come a long way to the point of where we now have ultra performance LC instead of HPLC. Still have that too. Mass spectrometry, which, you know, the methodologies, the 533 and 537.1 that Andrew described, are done by LC-MS. We also have precision chemistry, so that's columns, sample prep devices. In a different division than what I'm in, we also have thermal and material analysis, and then we have additional competencies in sample preparation, automated sample preparation, things like the Andrew robot that you see in the top right. And then we work in conjunction with ERA. They are a Waters company. Many of you might know them for proficiency testing, but we have been together as partners, and ERA, part of Waters Corporation. It has to be some 20 years at this point. And so those were an awful lot of information about the individual bits. For those of you that have not seen a workflow for PFAS, at least the analytical side, you have everything from your recovery and precision standard solutions, things that I'm not showing here, calibration standards, etc., your vials, your columns, et cetera, all the way through to the software, and then professional services that help implement the system and get folks up and running if they've never done PFAS in the lab. And then ERA with their certified reference materials and proficiency testing to test the capabilities of the folks running these methods, and make sure that you hold your accreditation in whatever methodology that you're running. And we'll go a little deeper dive into some of these as the program goes on here. So to bring everyone up sort of to a base level, I wanted to talk about liquid chromatography and mass spectrometry, but LC is the first part. You heard some talks about cleaned mobile phases. So those are the solvents that go through a pump or a solvent delivery system. You inject a sample, and then the analytes that are in that sample go into a column, and that column is filled with particles that the sample interacts with, and you see the different components will actually separate based on their interaction with this particle. So you didn't imagine that yellow, red, and blue are actually compounds in the HPLC column. But those things interact, those analytes on the right, if you look, that's what goes on in an HPLC column. The analyte interacts with porous particles, some more, some less, and hence you get separation. And back into the left panel, you see a detector pick up on each of those analytes, and you get peaks in what's called a chromatogram. So everybody's experts on LCs. Let's move on to the mass spec. Actually, in a second, let me talk to you about one key thing. You heard from Jennifer about the contamination that could be in the lab, and it will be nearly impossible to get rid of the contamination in some of the solvents. And the system, you replace all the wetted parts as best as possible, get rid of those Teflon solvent lines that supply your system with solvent, and put in an isolator column, hold up column, hold back column. That's to hold up all the contamination in the system prior to seeing your analytes. And on the right, it shows if that kit is installed. When you inject your standard, you see the standard, but there's no peak in the blank. So ultimately, that's what you're shooting for, is no background peaks in your solvent blank system, again, that you heard either Andrew or Jennifer talking about. And so you have that LC feeding the mass spec. Where it goes to first is called the ion source, and in this case, we're talking about electrospray ionization, or ESI, if you've ever heard that little acronym. You have a sprayer and some nitrogen nebulization gas, and you spray the gas at an inlet cone, and that cone draws the sample in, it desolvates, and then you look for whatever compounds, and in this case, charged ions come out all the way in the detection system. And so now everybody's mass spec is nearly up to date. We run the mass spec in what's called multiple reaction monitoring, MRM. And this allows for high selectivity because the way we run it helps to reduce and eliminate interferences. High sensitivity because we're spending time on only one compound. And accurate and robustness as well. So the example at the bottom left shows you a little bit about what this multiple reaction monitoring is. So let's take one of the better known PFAS that's molecular weight is 412, and we're going to put a positive charge on it, so hence the nearly 413. And then we're going to fragment it in a collision cell. Imagine taking a coffee cup and you bang it against the wall and you create little pieces, and then you can put it back together and see what the coffee cup was made from. And so we monitor 2 fragment ions, happens to be 369 and 169, and that's your quantitative and qualitative ion for confirmation. A bit long-winded and complex perhaps, but in the end, what you're shooting for is to get a calibration curve, and then when you run your sample, you monitor what the area count is, and you plot that against the cal curve, and you end up with a concentration. And so these are the calibration curves for method 533. Again, for PFOA, the C8, and then somewhat little smaller, everybody knows Gen X, HFPODA. And if you look a little tighter towards the bottom, you'll see that nearly everything falls within a line. And then you can use that calculation that you see at the top right of either of these cal curves in order to calculate concentration of your unknown sample. And in the end, this is what it gives you. You'll get a peak in the chromatogram. On the left are carboxylates, on the right sulfonates, and of course there are other PFAS, but I'm trying to limit what we show here. But it's peaks that come out, and they've all been separated on that LC column, and the mass spec has detected them, and they come out as a peak, and then that peak gets translated to a concentration based on your calibration curve. There are many methods that the EPA, ASTM, European Union, and ISO have put out. The 2 ones that you're most interested in, likely when it comes to drinking water in the States, is EPA 533 and 537. You all get a PDF copy of the slides, so these things that are underlined are actually hyperlinks, and it takes you to a particular sample prep product or a collateral or a particular recommended column in this case. And so this will be a handy table for those of you that are interested in both of the drinking water methods, but then methods outside, which might be things like influent or effluent wastewater, etc. The peaks, if you look at running these in drinking water, 537.1 on the left, 533 on the right. Again, you'll see some distinct differences. Some of the compounds retain a little differently. Some of the compounds you look for in one method may not be in the other method. And hence the development of 1633, taking the compounds in both of these methods and then putting them all together and then some for a non-drinking water methodologies. Andrew talked about this, so I don't want to belabor the point, but the National Primary Drinking Water Regulation, again, 4 parts per trillion limit on PFAS and PFOA. And you do have maximum contaminant levels, and you heard from Andrew on that. And then there's a 10 ppt level on several other category chemicals that you see here. And on top of those individual levels, that's where you have the hazardous index, and it has to be a mixture of 2+ of the above, including PFBS in this case, which is not monitored separately. So I know everybody's been throwing around numbers. Andrew certainly mentioned parts per trillion. And so a way to think about parts per trillion is, the equivalent of a drop of water in 20 Olympic-sized swimming pools. So again, concentration perspective there. Or at the end, you're distance 6 inches out of 93 million mile journey. I didn't put the quadrillion because one of the health advisory limits was in the parts per quadrillion. That's trying to find 4 parts per quadrillion, 4 ants in all of the Earth's population of ants. So that's telling you something. And all of this is really highly dependent, like you heard from Jennifer, on the how much caution you pay in the field, and especially how much caution you might pay in the laboratory as well. So the requirements for the two different methods, different compounds, many of which overlap between 537.1 and 533. Different sample preps. So one uses our set pack PS2 cartridge, and the other is an Oasis wax, so it's a weak anionic exchange. So two different mechanisms for retaining PFAS. I'm sure your lab or the labs you use have a preference on which one they want, but just to let you know they are a bit different. And then the hyperlinks at the bottom will take you to some application notes. So our mid-range product is a TQS Micro. It meets all the requirements of the National Public Drinking Water. It's very difficult to see here, but once you get the slides, you'll be able to blow them up or even print them out. That the method blank produces just barely a bump. Then the 0.2, 0.5, and then 4 nanogram per liter samples that you see here, and those are reported as sample equivalent concentrations. Again, one of the things in the methodology talks about recovery, and recovery being somewhere between 70 and 130%, if I remember correctly. And either the methods, 537.1 or 533, using LCM SMS, easily meet that method requirement. But what if you need to consistently get down to lower and lower quantitative analysis? So maybe you want better reproducibility at the lowest levels of quantitation, or you really do need to get down below that, single-digit part per trillion. There are systems that are out there that are capable of doing that. If you're, again, running 533 or 537, along with the sample prep, you can get super low, like the health advisories. Or if you're not tied to those and you want to do drinking water or non-potable water, soil sediment by something called direct inject, where you sort of bypass the sample analysis, then you're going to need something like a top-end instrument. So just to give you ideas, the previous technology, right before the launch of our high-end mass spec, it's called a TQ Absolute. Again, you're seeing sensitivity improvements anywhere from a factor of 2 all the way up into the 15, 20 range in area percents. Sample prep for direct injection, pretty darn simple. Water or soil, again, you'll take 5 mils of water or a couple of grams of soil, and a lot of those things that you see, you'll filter because you don't want to get particulates in the mass spec. That's a bad thing. Transfer things to a polypropylene vial, and then you're off. So that's one of the gotchas in the lab. You're going to stay away from glass vials or any kind of glassware. You're going to work with either high density polyethylene or polypropylene. Low density polypropylene or glass is going to really screw up the analysis, let alone your system. So that's one of the things to avoid, amongst all the other wonderful things that Jennifer talked about. Aluminum foil. I think she mentioned the HEPA filter on the lab HVAC. Pipette tips. Test those out. Test out all the other equipment that's going to be there. Everybody's going to be using nitrile gloves, but it's the powder-free version. Don't use the ones that are powdered on the inside. So loads of things that you have to be aware of. Cleaning agents that housekeeping might come into the lab and want to clean your window or something like that. Even the slightest bit, if you're getting down into these parts per trillion, you need to be cautious of. Having a separate dedicated area, doesn't have to be fancy. That's always a good thing. So again, we collaborate with quite a number of folks within the US EPA, as well as other facilities around the globe. And these are ground surface and wastewater samples that we've acquired for them that they've graciously prepped and sent us, sometimes even just sending us the raw waters, which I know those are fun to work with in the lab. And so some actual results from those water, some of which would pass fine. Some of the detection limits, you're down into the low single parts per trillion, some of which may not. So again, it's just real world samples showing up here. And the detection limits, and I really don't want to go through all of these, and the only thing I really want to point out is the yellow. It doesn't matter most days how clean you've kept yourself or the lab. Somewhere contamination is going to find its way through. Hopefully you can figure out where. Sometimes it just doesn't happen, and all of a sudden, you know, a typical thing like 6.2 FTS is going to show up as a background. PFBA, again, detection limits are high there because of laboratory background. It's the real world we end up working with. And this is that part per quadrillion that you heard from Andrew. UCMR limits in the low nanogram per liter, but some of the health advisory is just a whisper in the parts per quadrillion area. Very difficult to get through unless you're super, super cautious. If you're prepping samples, this is a solid phase extraction manifold that you see here. The basic system with a vacuum pump and waste, and then you end up pulling your sample through the cartridge, collecting the effluent, and then concentrating it. And then, you know, so you take, in the case for the reaching those parts per quadrillion limit, taking a 250 mil volume of the sample down to 0.5 ml is what gets you that concentration in order to run it on the TQ Absolute. You heard that this is everything is everywhere. I won't belabel the point, but things in the field, no waterproof clothing, jackets or rain gear, anything like that. Tyvek suits, people use them, but not the waterproof ones. Stay away from those. Sunscreen and insect repellent, don't do it. If you've had fast food, make sure you're scrubbed up for that. Certainly have a separate staging area for your samples versus if there's a eating area. And then in the lab, things we didn't talk about is, the solvent lines for the LC are going to be replaced with PEEK tubing or stainless steel. Stay away from deactivated syringes because you're just going to contaminate your samples. So that enough about all the contamination. We'll keep going and finish up. Education and fundamental training, we do at Waters to get your PFAS system going. There is, again, based on where you are in the world, but in the US, there's specific training for 533 and 537.1, and I believe 1633 as well. So if you want to start those up in the lab and you're looking at getting an LC-MS system from Waters, then of course we have certified people that come out and do courses, not only at Waters, but at your laboratory. Getting to near the end, laboratory accreditation. You have the EPA, they're the ones that set the rules and regulations. Everybody has to submit, formulations, et cetera, to them. They do a lot of the testing, a lot of risk analysis, and they're the ones that help promulgate those methods under the Safe Drinking Water Act, et cetera, when they come into law. State agencies, they're the ones that certify the labs. There's some other certification programs at the bottom that I list. And then the laboratories, you're certified in the state with your particular method of interest, accreditation, certification. to run those as a qualified lab. Most everybody in the States probably knows of NELAP accreditation program, and they have established and implemented a standardized program for accreditation of those environmental methods in certain labs. And then people also follow ISO 17025, and that's just a quality management system whereby testing standards and the way you operate within the lab with standard operating procedures take effect. So I mentioned ERA. Most everybody hopefully knows about it, but if not, they are a provider of proficiency tests and certified reference materials. The certified reference materials, they're known concentrations with a known uncertainty and stability, and the values are traceable along the way, and they contain a certificate of analysis. So that helps with method development or troubleshooting. For example, if you think you're in control of your method, if you run a CRM at the end where you know the concentrations, and then somebody compares your lab results to that, you'll know if your methodology is working. And this is an example of a 1633 result, but using the CRM from ERA and wastewater. You can see the blue line is your certified value, the gray line is the experimental value, and then your minimum and maximum certified values, and the experimental falls well within those lines. So it's a good test at the end if you want to confirm that your method is working properly. And that's one of the things that I think Jen said as well, besides running your method blanks. Proficiency testing, that's a mandatory thing for accreditation. Probably quarterly testing to continue on with your method validation and certification. Loads of resources available, both direct links to some application notes, also to some resources on our PFAS resource hub, application notebooks that are a compendium of all the PFAS testing apps that we have, case studies, infographics, and all about your sample preparation, etc. So like I said, we do a lot of collaboration with the EPA, UMass Amherst. Amherst is just right down, I say right down the road a piece because I'm from Pennsylvania, but it's just right down the road from our Milford, Massachusetts lab where the folks that are listed at Waters Corporation mostly reside, but there are a few, the two Stewarts work remotely, one in the UK, one near the Cincinnati, Ohio area. So thank you all to all the help, and thank you all for listening in. And at this point, I am going to switch over to Kyle, who will talk a little bit more about the data. Kyle, it's over to you.
Cool, thanks, Ken. Hi, everyone. My name is Kyle Power. I've been with Trihydro for almost 12 years now, and I'm in manager and been doing data validation, our chemical data evaluation services at Trihydro. And mainly what we deal with is laboratory relations and technical support for anything chemistry related, database management, anything data validation. And so this was a good topic for us to cover when it comes to PFAS, because there's a lot of interesting things with PFAS, but also a lot of different things too. So next slide. Okay, so my portion of the talk is how to get the best data from the lab. And, Jennifer and Ken kind of went through a lot of the lab stuff, the things that they're doing to maintain the best data, best results that gives you that level of confidence. Now, there's a bunch of other things you can do outside the lab to get there as well. And so I have a couple topics listed here. One is proper communication with your client, agencies, regulators, laboratories, consultants. And mainly this has to do with kind of setting up the project or the site so you know what's going on with PFAS and kind of all the preliminary things before it reaches to the lab, and all of the things after the lab that you have to do when it comes to your data, and that includes data validation. I think that's a very important step with any piece of data you get from the lab. Other things you could do, possible audits. This is something that Trihydro does and we've done in the past. Mainly checking that those things are being done in the laboratory, the cleanliness, the procedures, that they're running the methods properly, they're doing all their checks, they're doing all their quality control samples at the frequency they need to. Mainly, are they meeting all of their laboratory operation requirements that are established by the EPA or whatever accreditation that they're going for. Next slide. So the first point of having proper communication between all parties involved, I always like to bring up a project plan, specifically a quality assurance project plan, or a QAPP. And for those of you who aren't familiar with what a QAPP is, it's essentially when all parties involved get together and they establish pretty much all the rules when it comes to the life of a project or a site. And so you have to think of things like, why are we on site? Are we cleaning up some things? Are we trying to maintain compliance for a site? What's going on? Why are we there? And how do we meet our end goal? And so, like, how often are we going on site to sample? What network are we using? How are we sampling? What quality control samples? These kinds of things that we have to discuss between all parties. And, a lot of people just consider, oh, I'm a consultant. Maybe I only just need to talk with some regulators or agencies or that kind of stuff. But a lot of people don't consider the lab in this, but they're a very important part of this process because they're the ones that are going to be taking your samples, running them by these specific methods, doing all of these things. And so you have to have all parties sign off on what quality control you're trying to meet. And if that isn't met, what do you have to do? Are there repercussions? Do we have to resample? Do we have to reanalyze? All of these things can be mapped out in a quality control, quality assurance project plan, and in great detail too. Sometimes I've seen plans where it's not too detailed or they're referencing a lot of other guidances. I've seen some that are very custom and they have specific detection limits that you have to meet, specific, quality control frequencies you have to meet, all of this kind of stuff. And so I have a list of a couple of things to consider when putting together one of these QAPPs. First of all, what guidance are you considering that's out there? Are you considering a guidance at all, or are you kind of building this custom? I always like to look to see what kind of guidances are out there that have been developed either by the requirements in the method. Department of Defense has a lot of stuff out there for their specific sites. Are you going to use both? Are you going to take pieces and parts of that? This is a conversation that you have to have with your client regulators, depending on federal level, state level. There's a lot of different stuff out there, and PFAS is one of those things where there's a lot of information out there, but I've heard the term the Wild West thrown out there, where a lot of people it's kind of unknown, and people are doing what they want to do, and that's okay in some parts, but sometimes those requirements need to be established, and you need to put those in place and to meet MCL goals, that kind of stuff. Other things to consider: field sampling methods. Jennifer and Ken went through a lot about the contamination aspect, and so what things can you do in the field. I've heard anything from you can't wear makeup, you have to wear a full suit, you can't eat lunch, you can't do this, can't do that. And so establishing how you're going to sample your water or soil or whatever on your site, that's very important. It's a good thing to have in your QAPP document. Other field quality control samples, these are like your equipment blanks, field blanks, trip blanks, that kind of stuff. Blind duplicates, to test the precision of the lab. What types of these are you going to use, and what's the frequency you want to use? I've seen people not use any equipment blanks whatsoever. I've seen them use one per day. I've seen them use one equipment blank per sample, per location. That may seem a little bit overkill, but if you're worried about how you're collecting your samples well to well, it may be worth doing that. And so these are some things that you most likely have to test out for your specific site, and establish that early on in the project life. Other things to consider in your QAPP, since the laboratory is a part of this, what quality control samples are they running? Do you want them to run matrix spikes? They should be running laboratory control samples, method blanks. Do they have specific surrogates, internal standards, extracted internal standards? Are they running duplicates? Do they have any, detection limit studies? All this is important information to know when establishing this document, so you know what you're going into before you even go out and sample. And once this data is validated, how is it going to use for compliance review? Or based off of your data validation, if you have qualified data, how does that qualified data affect any decisions? If you have rejected data, hopefully you shouldn't be using rejected data, but all of those things should be established in that project plan. So this is, I think, is a very good document that every site should have, or at least consider. Next slide. Okay, so the second point on that intro slide was regarding data validation. And I'm going to go into what to consider during your procedures and typical things that I've come across and my team's come across when we've, validated this PFAS data over the years. So again, thing you should consider is what guidance are you following? Because, currently there's a couple out there. We have Department of Defense that has pretty detailed stuff. Method 537 has their own specific guidelines of what to look for. With other types of sampling, like volatile organics or semi-volatiles, metals, the EPA has established national functional guidelines that are out there, and they are consistently updated and revised. And really, we don't have much of that currently. I mean, I think that document is coming, and I think there's going to be, a very good need for it as we continue on with PFAS. It's not getting any better. And so, these are documents you should consider or, plan on using, in my opinion. And, at Trihydro and my team, we kind of use a combo of these. We get together, we discuss all these documents and guidances, and we kind of use our professional judgment as chemists and kind of see what works, what doesn't, how we should be evaluating data, how we shouldn't. And these are all things that we can discuss with project team members when we plan out our QAPP. And so, are you using one document? Are you using the other? Are you using both? Are you using neither? It's kind of up to discussion and up to debate. But I just want people to be aware that there are documents out there, and we should be using this validation kind of approach to data, because obviously the lab's going to run their samples and do their thing and try to keep as clean as possible, run their frequency of quality control samples, but there always has to be that confirmational level of evaluating that data, so we don't just take that as absolute. We have to take it a step further and evaluate it and see if we can actually use that data and make the appropriate decisions for our sites. The other thing to consider is what level of validation. The EPA has their specific stages that they like to evaluate. I've seen different types of validations that's been discussed based off of the lab talking. Myself, we use specific tier validations, but just explaining and putting this in your QAPP document is really important, because what level are we actually doing? Are we just doing like a level 1 where we're kind of just making sure that all the frequencies are met and everything in the QAPP is good to go, and we're not actually qualifying data? Or are we taking a step further and doing like a stage 2A where we're actually looking at the limits based off of high or low bias? We're actually qualifying that data, high or low. If we have gross exceedances, are we rejecting that data? And then there's even further validation levels where you're actually going into the instrumentation, the raw data, and making sure that the lab's doing their calculations correct. And so the extent of validation could get pretty lengthy, or it could be, not much at all. But, again, these expectations should be established in your QAPP and discussed with your lab to say, hey, this is the level of validation I'm going to do, just to let you know, I'm going to scrutinize this. I'm going to be scrutinizing this. I'm going to be lax on this. And so all parties know what's going on. Next slide. Okay, so, I've spoken with a lot of people who are data validators or chemists and kind of look at this data. Lab send you this report, and it's a lot of information. And this isn't just with PFAS. This is with all analytical data. But we've seen years worth of data, and we have come across some typical findings when it comes to PFAS. The first I'd like to point out is with the extracted internal standards. And so, uh, you know, good definition there. These are, related to, if you compare them to other methods and their internal standards, they're a little different. You extract them before, go through the process, all that kind of stuff. But, usually in other methods, you have, a couple internal standards that a representation of specific analyte groups. And, PFAS is interesting because based on your analyte list, you could have a designated extracted internal standard per analyte, and how they behave. And so really there's a lot of data that's generated based off of your list. And I bring this up because when we go through our data validation processes, it really depends on what guidance you're using. I have an image of Department of Defense section here when it comes to extracted internal standards. They go by a 50 to 150% recovery, limit. Laboratories, if you look at lab limits, those are going to be different, most likely. Sometimes if it's told by the lab and explained in a QAPP and that's a requirement, then you'll see the 50 to 150. But just be aware that depending what lab you use and depending what method they're using, they may be giving you the laboratory limits. They may be giving you your QAPP limits. All that should be established. But, if you consider laboratory limits, EISs, they tend to be within range and okay. But when you start considering 50 to 150, and even the stipulation of, extremely low area counts of less than 20%, that could lead to rejected data for non-detect values. And so these are things that you should be thinking about and looking at in your lab reports. And so that's a general finding that we see in EIS is usually leading to some qualified data, whether that be estimated data or rejected data. And then I also, have a list of some PFAS constituents on this slide. Those are, typically lower than you would when it comes to that 50 to 150, usually lab limits there tend to be fine, but those are common ones that I always see that are low. So just something to be aware of, that some compounds are affected more than others, depending on your method, how you're setting all the instrument up and all that kind of stuff. Next slide. And I also I always like to point out holding times. Based off your method, your holding times are going to be different. I list method 533 and 537 here. If you're considering outside drinking water, if you're considering, the 1613, that has different holding times that you consider based off of, how you preserve the sample. That's slightly different. But I always like to point out holding times because when it comes to other methods, this is generally the most common reason why we're rejecting data, or we're qualifying data, is we miss the holding time and we can't rely on it. These methods are slightly different when it comes to the guidance that are out there. Rejected values, if you miss the hold time, you're not immediately rejecting it, right? I have a couple of screenshots of some of the guidance that I found Department of Defense and the method 537 of, if you are outside your extracted holding time, your analysis holding time, usually just qualifying the data as estimated. That could be a good thing, that's not rejected. It could not be a thing at all, or based off of what you have in your, what you have to do for your site via your QAPP, it could be a big thing. And so just considering these holding times, it's always great to communicate with your lab, to make sure everyone's on the same page, to have it in your project plan documents. But this is something I always like to highlight when it comes to validation, because holding times are always an issue, especially when there's a lot of samples being filtered through the lab. They have other clients besides you, and so they're some labs are analyzing a bunch of samples every single day, and are consistently moving around their how they're analyzing, and prepping those, and so that could be an issue that you have to consider. And another point, consider the multiple guidances that exist when it comes to this, because you never know. The EPA could come out with their national functional guidelines of just PFAS in general, which could be completely different than the individual methods. That could be completely different than this Department of Defense guidance. So just be aware that some of these things may change over time as well when it comes to that validation. Next slide. And this is my last slide I have. Just other data validation considerations that you have to, that I always look at during our validations. And some highlights that I can point out is, I mentioned with the new 1613 method, the sample temperature, how you freeze that, how the lab stores it at a certain temperature can change the holding time, can change how they do things. Consider blank contamination. How are you treating those detections in the blanks? Are you, when it comes to validation, are you qualifying your data as, oh, there's a contamination, so it's non-detect, kind of thing, or are you using a 10 times rule? There's a lot of guidance out there that kind of you can go by and consider or not consider. Field duplicate, precision, what percentage of RPD are you going by? Are you going by a specific method? Are you going by a specific guidance? These are all things to consider. And then some other common ones that you have to look for when it comes to PFAS, very similar to other types of methods, your laboratory control samples, your matrix spikes, your calibrations. I'm not going to go into detail of numbers when it comes to these validation criteria. They're listed out in the guidance, but there's definitely a lot of, terminology in those guidance that allows professional judgment by a chemist and by the lab. And so those probably need to be discussed too if you're worried about some of those sections. Next slide. So yeah, that's kind of just an overview of things to consider when you want the best data from the lab, and when it comes to data validation, how you prep for that before you even start and going out and sampling PFAS. So there you go. I'll pass it off.
Great. Thank you to all of our speakers today. Lots of good information. Andrew, do you want to address the questions in the chat first?
Yes, Amy, that'll be appropriate. And actually, we received some excellent questions, very much in line with what the intent be for this sort of discussions. So invariably, these questions are to all of the panelists, and we can have an interactive methodology to addressing them for the benefit of the audience and, of course, the person who asked the question. I know that this was an extended time webinar. We typically have these 60 minutes, but this is an hour and a half. So hopefully the person who did ask the question is still present and would get the direct benefit of getting answers. And if for some reason the person is no longer available, we'll circle back with the individual and the group as a whole, so that again everybody can learn what the thoughts are on this. So the very first question came in. And this is very appropriate, because if you do any risk assessment whatsoever, the issue of significant digits in your hazard index, hazard quotients, and ultimately the risk decision is an extremely important one. So luckily, the answer is on that. So how many significant digits essentially in your hazard index? Because that may or may not impact the outcome. So luckily, there is an EPA document that was produced as in terms of guidance, and the answer is 1 significant digit in all your calculations for the hazard index. And then in the chat room, I've included the link to that document. So pretty direct question with the direct answer and EPA guidance substantiation. So if the participants still on the call, if you wanted to chat back, or if there's a way to unmute yourself, make sure that the answer provided is what was anticipated.
Andrew, right now the mute option is not available. So we can always provide email addresses in our follow-up. So if people have further questions, we can address them via email.
Thank you. So I would like to thank Nancy Harris for asking that question. And again, we stay interested in providing all the feedback that you would need. So thank you for that. The next question, and this is increasing complexity, and this question came from Paul. And the question is as follows: is there a confidence interval on analytical results? In other words, how accurate are the PFAS results? And this is a tremendous question, because this was the whole premise of having discussions like this. So since I am not a chemist by training, I'll defer that question to perhaps Ken or Kyle. And failing that, maybe Jennifer, if you all want to dispatch the answer for that. So the floor is open.
Well, I'll give a quick crack at it, and it should be one of the calculations that the lab does. We talked about, error bars in whatever the result that you give, and it's I believe going to be based on the accuracy of the calibration, the standards, whether or not there's been any contamination, but there should be a concentration error bar around the final result that's given, and that's going to be laboratory dependent on, everybody's going to be injecting samples several times, so that you do have that, relative standard error that you can report around each of the concentrations for whatever PFAS is found. So I do believe that's a method. It's highly dependent on the method and the results that they're getting for repeatability of injections, et cetera. So hopefully that has a good idea, if that's what you mean by confidence interval. It relies on the precision, how many times you can repeat it at a precise number, and then how accurate are your results versus a standard curve or a standard known concentration. And anybody else is welcome to chime in too. And if that doesn't answer the question, I know people that can give you a better one than I did.
Waiting for a couple seconds to see if Kyle, Jennifer Hatfield, and maybe partly due to some technical difficulties, we failed to introduce Jennifer Hatfield, who happens to be the at one time she was the head chemist at the 3M Corporation. So Jen is quite familiar with the challenges associated with testing for PFAS. So I don't know, Jennifer, if you have any comments on that or provide additional background or context.
Well, you've come off of mute. Nope, you're back on mute. And I know the trouble we can have with Teams, like everybody does, I'm sure, between connectivity and the
I
system
should be
function.
unmuted.
Yes, you
Yes,
are. There you
thank
go.
Oh, good.
Very good.
Oh, good. What was the question again?
So we were talking about accuracy of results, and the type of expected errors that a laboratory might see. Being concerned about the results that you might get. What's the plus or minus standard error that you might expect to see with a particular methodology.
We used to think that try and shoot for around plus minus 10%. And that seemed to be, we seem to be able to hold that fairly regularly. Sometimes it'd scream up to about 15. But, we've got a question coming up on calibration curve graphics, in straight line. The older mass specs, as these curves got bigger and bigger, started going quadratic. That's just a function of the instrument. And so, you start getting a little bit off at one end or another, and then that affects how accurate your instrumentation is. So rather than worry about how accurate the information is, we used to set, we want to be plus minus 10%, or as part of our we run everything GLP, so it's similar to what everybody else did, but we needed to be plus minus 10%. And or better. And so if it got out of control, we had control samples, we'd have to rerun, we'd have to redo things all the way up to set that experiment up again. Yeah, I was in environmental lab, which was one of about 7 or 8 labs at 3M that I was kind of the chemist on. So yeah, 10% was pretty okay. And if you want to get down to super duper low numbers, it starts getting more and more difficult. So 10. Do you agree, or do you think that's fairly close?
I like the answer. I know there's been a couple of questions, not only on the confidence interval, but the linearity versus quadratic. And I know if you go back to the first slide that had, and you don't have to do this, Amy, but if you go back to the first slide where I showed the first calibration curve, that was expanded to the whole range, and it looked like, but it's not. It looked like it hit the XY axis before the zero. That's an artifact of the display. In fact, 533 calibration curve could be linear or quadratic, but it has to be forced through the origin. So if you look at the second one where it sort of explodes ties into the bottom half of the calibration curve, you can see that it actually is forced through zero. And that's to help give a better estimate of background levels in the particular method. And so the repeatability at the lowest level, the percent deviation is listed in the EPA methodologies. There's, in fact, that I'd recommend if you haven't looked, just Google EPA method 533 and you'll come up with the methodologies, and it talks about all the calibration curves, the continuing calibration curve, isotope dilution, because you're putting in isotopes of the native compounds, and which will have the same retention time, and you're doing the calibration and adjustments to the calibration based on the area percent of the isotope standards that are in there. And then there's surrogate chance standards to test everything to make sure that you're on board. So there's a lot to the answer about beyond what is the accuracy at the lowest level in that case. And then if we need to go further into detail about all of that, again, we can get you in touch with the people that know more about the calculations and the methodologies that do this day in and day out.
Thank you, Ken. So you invariably answered the question that was presented in the chat window. And before we need to proceed with closing remarks and statements, I do want to maybe ask you directly, what does the method require in terms of the separation of delta between the method detection limit and quantitation limit? So I've heard some numbers thrown out, but can you elaborate on what the methods require you to see separately on that lower end of the calibration curve?
Yeah, that I'd have to get someone else to answer because I'm not quite sure I'm understanding what your question actually is. Sorry about that.
Practically the difference between the method detection limit and quantitation limit has to be X. So is it 2? Is it 10? What the
I see what you mean. Usually you want to go, if your quantitative limit that you have to give somebody, let's say it's 4 parts per trillion, typically you need your method to be able to quantitate half of that, so down to 2 parts per trillion, so that you can actually accurately report the 4 parts per trillion. And then the method detection limit is going to be even below that, and that will be determined sheerly from what your background levels are. So everything that we talked about in watching what you do in the lab, how well you collected the sample versus the blanks that you've collected along the way, the laboratory blanks. So that's really going to affect what your limit of detection, and possibly if you've really contaminated, it's going to be your limit of quantitation. As you saw those yellow highlights from one of the experiments that we did, there was a lot of 6.2 FTS in the background. So that's really what's going to limit your quantitative and detection limits in that case. But usually shoot for 50% of half of what the quantitation that you need to do for that particular method. And a lot of that I think is even stated in the various EPA documents.
Very good. Ken, thank you for that. And before hand it back to Amy to thank the speakers and the audience participating, just want to let you know this is not the only webinar on this issue. We intended to have a multi-part series when the next event will dive much deeper into actual data for water, drinking water, and we'll take a step-by-step methodology, how it would look like if we have to force these data through various approaches, QAQCs, and also what the compliance outcome will look like. So stay tuned for an invite. And then, Amy, thank you. Go ahead and if you could close us out.
And one second before we leave, just to make sure that I correct myself, because I deal globally. I think Europe is half the method detection limit, where the United States may be 1/3. So I'll have to double check, but that seems to make maybe a little more sense in my head if I think correctly. So
Thank
now,
you.
Amy, go ahead and take us away.
All right. Well, thank you to all of our presenters. As mentioned, we will be providing a recording of today's webinar to all of those folks who have registered. If we didn't get to your questions in our time together, we will answer them via email after the webinar. And thank you so much to all of our presenters for that wonderful information, very valuable, lots of good information here. And stay tuned for details on the next webinar in our PFAS series. So thank you, everyone. And I'll open the floor up to you guys for last minute comments and thank yous, and then we'll wrap it up.
Yeah, everyone certainly have a
Thank you.
fine rest of your day, whether it's the rest of the afternoon or evening.
All righty. Take care.
Thank you.